Good afternoon, everybody. I found this to be a fascinating topic, and the more I researched it, the wider it got, and hopefully I can cover it as best as possible in 15 minutes. Here are my disclosures. None of them have any bearing on this presentation except for the Reshape Life Sciences, which created the V-Block device, which I will describe the data. It became obvious to me in looking up this topic that the central nervous system has quite a bit of control over metabolic processes. You know, we talk a lot about GLP-1 and ghrelin and operations, but we don't talk a lot about what the actual nervous system does. And there's now a growing body of literature and studies that electrical manipulation of the central nervous system via the vagus nerve, via other nerves, via the brain, has profound effects on the metabolic disorders, and possibly in the future might provide for us a less invasive way to treat these diseases. So let's just take a roundabout view of it. First of all, these are only some of the processes that the CNS controls: appetite, body weight, energy balance, blood sugar regulation, but there's probably others that we're first scratching the surface. I don't need to once again reiterate metabolic surgery. I think Dr. Suter and I were just making the point that it's been around a while as a term, and that Henry Buckwald gets credit for at least publishing the term back in a book he wrote with his partner, Dr. Varco, in 1978. Well, you'll hear a term used called neuromodulation, and I just wanted to define it. It's using an electrical pulse generator, which is essentially a pacemaker, and connecting it to some aspect of the GI tract, whether it's nerve, whether it's muscle, whether it's adrenal glands, and either stimulating that pathway or interrupting that pathway completely. There are a lot of other things it may be doing. The more we look, we see it may involve hormones, etc. But the one thing we're not doing is pacing it. It's not the heart. Well, the very first one of these was almost 20 years ago, which was the implantable gastric stimulator, or the IGS. This was a pacemaker-like device that was on 24 hours a day, 7 days a week, never took a break, and it was connected to the lesser curvature of the stomach near the nerves of Latarjet. Extremely safe, very few complications, caused essentially no symptoms in patients. But unfortunately, at the time, after 800 patients in different studies, the data was not conclusive enough for this to go forward. The FDA never approved it. However, there were patients who responded extremely well to it, and some of them had a better than 35% excess weight loss with this device and nothing else. We don't really know what the mechanism of action was, and the company that was involved with this device ran out of money, basically. It was bought by a larger company who's one of our sponsors here. They'll go nameless. And they buried it, although my understanding is they are getting good results in their laboratory work, and this will likely appear again. But it already has, actually, in the form of other devices, which I'll explain in a minute. But let's talk a little bit about mechanism of action. This is a stomach, and what you see here is a big, basically manometer in the stomach. We inflate that, and it can measure the pressure of the top of the stomach, the most thinnest part of the stomach, the fundus, which normally will distend after we eat a meal and tell us, you know, you're done eating, don't eat anymore. Well, if you put the IGS on, this increase here is the normal increase you see with eating. But if you put the stimulator on, you get a much greater distension of that gastric fundus, and this is possibly one of the many mechanisms that's been talked about and studied. How exactly does this work to cause satiety? This was the very last trial using the device. This was the SHAPE trial, and unfortunately, the trial, which was well designed, did not take into account that a lot of the devices at the power we were stimulating was going to run out of juice. Mainly they were dead batteries. And it turned out that there was no statistical significance between the control group and the active group. This was a multi-center randomized trial. And that was basically the end of this device for the near future. But once again, it's being looked at on a lab work and animal work, and it may come back. But like most other designs, somebody's going to come along and make one a little bit better. And this is the Medicare device, which is a device that's unique in the fact that it only goes on after eating. So in the rest of the time, it's off. And by perceiving eating and then activating, it may prevent what we think might be compensatory mechanisms that may form when something is running 24/7. Their data was very interesting for metabolic diseases. They showed actually statistically significant benefits for hemoglobin A1C, for liver numbers, etc., and with essentially very mild weight loss. There wasn't much weight loss, so clearly more metabolic effects than weight loss effects. They then did this crossover study looking not at weight loss, but at diabetes, and found that those patients that were in the control group the first half of the trial, 12 weeks, and maybe were getting a placebo effect or whatnot with their hemoglobin A1C, but once they crossed over to the active group, they continued to drop their hemoglobin A1C, with a group that was active and now was turned off actually had a rebound of their diabetes. Just interesting. Not really sure what to make of all this. Very, very small numbers. And there was yet another device, the Ability device, that was looked at in different parts of Europe. One of the people here, Luca, was involved with it. And basically, this was also meal activated, but it included an accelerometer to sense physical activity. And then you could reward patients for good physical activity, and you can reprimand them for poor activity. Tom Horbach in Germany did the first trial. It had some nice weight loss, reduction in appetite, improvement in physical activity. And you look at this and you say, dynamite, we're there. But you know, you always have to read a paper carefully. There was a 1 in 5 withdrawal from the study prior to the conclusion, and only 31 patients completed the 12 months and very few beyond. So there was quite a drop-off in patients in the trial, and I think that has to be kept in mind. Luca Brachetto did a follow-up trial with it, and his results were also reasonable, but I don't think this device has made it beyond this study, which was a couple years ago, and we'll see if it comes back at some point, because it is very, very safe, it doesn't cause a lot of symptoms, and it may be effective for the metabolic disorders. What if we move down the track a little bit on the duodenum? There was a study performed by putting electrodes in the duodenum that demonstrated improvements in blood sugars in an animal model, and it did so by delaying the emptying of the stomach after an oral glucose tolerance test, increasing duodenal flow so that the chyme in the small bowel and the stomach passed through faster, reduced absorption of sugars and fats in the small bowel, and increased the sugars and fats in the stool. At the same time, insulin levels, instead of increasing with the glucose load, were dropping compared to control. This was an animal study, and the results here show with the control, you had a bump in the glucose after glucose tolerance test that was blunted in the stimulation group, and the insulin levels were blunted as well. So the sugar went down and the insulin went down at the same time. This demonstrates the gastric emptying was reduced when you stimulated the duodenum. And this demonstrated that glucose absorption was reduced when you stimulated the duodenum. So instead of somehow dealing with the sugar in the bloodstream, this was preventing sugar from being absorbed. This was another study looking at intestinal stimulation and demonstrated in at least a rat model that you can reduce food intake and body weight by stimulating the small intestine. More out of their same group, they were able to demonstrate that blood sugars were lower in the animals that were stimulated, and that the insulin resistance also dropped. This is the sham group, this is the on group, and you could see they were like basically same as the control group, and just more of the same. Now, what about the vagus nerve? The vagus nerve is a very interesting nerve. It's the 10th cranial nerve. It's the longest cranial nerve in the body. And in fact, it's not two nerves, one on the left, one on the right. It's actually one nerve with two trunks. What's even more interesting, it's broken down unevenly between efferent and afferent. 85% of this nerve is afferent, sending signals from the gut to the brain, and only 15% or so are efferent, which is the vagus nerve in the brain directing the GI tract. And most of what happens with the vagus nerve with directing the GI tract has to do with gastric emptying, changes in food regulation and hunger and digestion, but also keep in mind that the vagus, for the hell of it, has a few other uses as well. The afferent nerve function is what we're most interested in because that basically is the whole story about body weight and metabolism. And weight regulation by the vagus nerve is multifactorial, as you can see here. And again, I'm pushing a little quickly for time. I want to make sure I get through all this. This is just sort of a very wide topic. We've demonstrated that when the vagus nerve is blocked, whether it's cut or whether it's blocked with electrical stimulation, what you actually can do is get a reduction in appetite. And the drop in appetite was very interesting because in this study, the humans that were studied in this experiment had a proportional drop of protein, carbohydrate, and fat, which suggested they ate less of what they normally eat. So they weren't going to any sort of operatively, and it was 56% here. It was reduced, I'm sorry, 56 patients. It was reduced down to 28 patients. It was cut in half. And the numbers at 24 months were almost half as well. So half of the patients that were pre-diabetic were no longer pre-diabetic at 12 and 24 months, and half of the patients with metabolic syndrome no longer had metabolic syndrome at 12 and 24 months. The mechanism of action, once again, we have a number of different theories. This is data from someone unrelated to the trials, and what he found in a group of rats using the V-block device was that it seemed to have no effect on gut hormones, and I use just CCK as an example. It did seem to affect the brain with gene expression, and that was quite interesting. By stimulating or blocking the vagus nerve, you're changing gene expression in the brain. And it also seemed to have effects on another one of the brain genes. What about sticking electrodes deep into your brain? Well, I don't know that people would choose that over a sleeve gastrectomy. I probably wouldn't. But it's been looked at in different ways. And if you stimulate different parts of the brain, you get different responses. This was a group of patients with Parkinson, and normally they lose weight with time as the disease gets worse. In this case, it maintained their body weight. If you put the, almost done, if you put the stimulator in another part of the brain, it created weight loss. Same here. This is using alloxin to knock out the beta cells to create diabetes, and if you stimulate at the same time you give the alloxin, the diabetes is dramatically better than it was in the group that was the control group. So here are the issues with neuromodulation. We really don't know how to use it yet. We don't know how to set the device. We don't know where to put the electrodes. But if we ever do figure that out, and we figure out the right parameters to set it, I think we may have something here. It's just a matter, it's going to take a fair bit of work to figure all this out. So the CNS has several roles in regulating metabolic processes. Electrical stimulation, or I should say manipulation, because it could also be blocking of the CNS, has a lot of promise for the treatments of a wide range of metabolic disorders. But we're going to have to put our thinking caps on and figure some of this out and do the appropriate studies. Thank you very much.
Thank you.